Hello, everybody. I'm Kevin Outterson. I'm Executive Director of CARB-X. Welcome to today's webinar. World Antimicrobial Awareness Week begins tomorrow. So what better way to kick that off than to highlight how CARB-X protects humanity from the most serious bacterial threats by accelerating antibacterial product development. Since CARB-X was launched in 2016, we have contracted 72 projects, and we should be announcing 73, 74, and 75 in the near future. We currently fund and support 46 innovative antibacterial projects as of today. Erin Duffy, CARB-X R&D Chief, will tell you a lot more about our pipeline, priorities, and progress in a few minutes. Given our time constraints today, we had a truly impossible task, how to select just 3 out of those 46 great projects to present today. There are many highly innovative projects that you won't hear from today. You can check them out on the portfolio section of our website. But I'm delighted to hear today from Eligo Bioscience, Peptilogics, and Vedanta Biosciences. We have invited the CEOs of those companies here to tell us more. And because CEOs love to talk about their companies, we've given them a strict time limit. So thank you to Xavier Duportet, to Jonathan Steckbeck, and to Bernat Olle for joining us today. Before we get started, I want to say a few words about CARB-X for the coming year. CARB-X is now in our 5th year of the original 5-year BARDA award. Our other funders stretch out into 2021 and 2022. CARB-X funders have committed $480 million in non-dilutive funding, which has made everything that we do possible. We've spent about $257 million of that on open global funding rounds in the last 4 years, signing the 72, 73, 74, 75 milestone-based funding agreements, plus 12 active projects for which we have already extended funding and support through an option after milestone science-based review. We anticipate that many projects in our existing portfolio will hit milestones. That is the goal. And we'll request the exercise of options to carry their programs further all the way to the end of their phase 1 work from CARB-X and on to graduation from CARB-X. Because of that big chunk of work in the future that we're committed to as well, as we move forward, we are focusing our current resources on supporting the 72 projects already in the pipeline and those that are currently under negotiation. We are discussing with our funders, of course, next steps on additional funding for CARB-X, but the launch of any new funding rounds will be dependent on the successful conclusion of new funding. So now, please let me introduce Erin Duffy, who will provide an update on the CARB-X portfolio. Erin has served as the CARB-X R&D Chief since September 2019. She's been responsible for managing the global portfolio objectives and leadership of the entire R&D team. She's a member of our CARB-X executive team and also sits on the Joint Oversight Committee, which is CARB-X's governing board. She's an accomplished life sciences executive, former Medanta Therapeutics Executive Vice President, Chief Scientific Officer, and before that with Rybex in the same location, and head of the company's New Haven research site. We're very grateful to have Erin Duffy with us today, and I'll turn it over to Erin.
Thank you very much, Kevin. And from me also, welcome to everybody to this webinar. I'm looking forward to hearing what our CEOs have to tell us about their programs. Next slide, please. So Kevin will have already said much of this, but I'll re-emphasize that since inception, CARB-X has funded 72 projects. And I think in the upcoming days, as Kevin said, you're going to hear about 73, 74, and 75. So certainly stay tuned for that. Of those 72, 4 are currently active programs at all stages of development, and I'll show you that in an upcoming slide. We represent 10 different countries in these 46 active projects, and we've had 7 project graduates. What does that mean? That means programs in our therapeutics and prevention space that have crested successfully their first in human programs. Actually, Vedanta and Bernat are an example from our prevention portfolio of that. In our diagnostics portfolio, a graduate means one who has completed verification and validation and is on way to launch of a product. Next slide, please. So everything in our portfolio begins with a laser focus on the high unmet medical need. And when we think about pathogens, we focus both on the CDC and the WHO lists. And as you can see here, the majority of our programs target either the critical or urgent sets, and then the serious or high sets, with very few in the concerning or other categories. So a laser focus on the key pathogens for which we don't have good therapies today. Next slide, please. When we look at our therapeutics and prevention portfolio shown here on the left, you see that we emphasize high unmet medical need in terms of syndromes or indications. And so if you start around 12 o'clock and go to the right, we have a large emphasis on lower respiratory tract infections. And if you think about it for a moment, the last 10 antibiotics since 2010 that have been approved, 4 of them have an indication of HAP/VAP, 3 of them community-acquired bacterial pneumonia, the rest for acute bacterial skin. And so there remains a need not only for that particular syndrome, but also for pathogen coverage therein. Beyond that, you see an emphasis on bloodstream infections or sepsis, obviously a high area of unmet medical need. And then next to that in the gray, you see urinary tract infections still requiring new therapies, particularly with an emphasis on performance characteristics that bring oral options for one, as well as, again, the treatment of drug-resistant organisms. It's important to note that we do have a diagnostics portfolio, and our emphasis in there is not only in bringing the best technology for the different syndromes, but importantly, marrying up or aligning with our therapeutics and prevention portfolio. And this is both for the conduct of clinical trials as well as for ultimate market uptake. You can see just by the colors that the emphasis is very similar to that in the therapeutics and prevention portfolio. Next slide, please. So when we think about our therapeutics portfolio, certainly there are 2 categories. There's the category on this slide, which emphasizes validated mechanism of action, largely with new chemistries. And in that little sliver from about 11 to 12 o'clock, truly novel mechanisms of action or inhibition. So certainly we have programs that target cell wall, lots of them, both non-peptide and peptide class. We're lucky today to have Jonathan from Peptilogics to talk to us about their locally applied peptide program focused on prosthetic joints infections. We also have programs that target DNA synthesis and protein synthesis, many different mechanisms of inhibition in those mechanisms of action are represented. As well, we have fatty acid biosynthesis programs that come in a focus on many different pathogens. Next slide, please. But our therapeutics portfolio also emphasizes many trailblazing, what we call non-traditional programs. So again, if you start at 12 o'clock, we have many antivirulence programs, including one, FIM-H, shown in bold here, which is a GSK program recently entering the portfolio that has commenced first in human studies. We have non-beta-lactamase inhibitor potentiator programs. We have pretty innovative immune directing programs and antisense programs. We have some protein programs, both systemic and locally applied, antibody programs in therapeutics and also in prevention. You'll see that on the next slide, and even phage programs for treatment. We have something that fits into the category of truly novel, and this is a nanosponge program from Celix that also recently entered the portfolio. Next slide, please. Our prevention portfolio also emphasizes multiple modalities. So again, starting at 12 o'clock, a large portion of our portfolio is vaccines, but vaccines for AMR, not a lot of those. And our programs are focused on areas where there have been a lot of failures, but we have some really exciting programs, and we're hopeful that they will make it through, as well as pathogens for which we haven't had vaccines. And so Klebsiella pneumonia is a key one of those, and others. We're very excited to see those progress. We have microbiome-based programs, and today from Vedanta, we'll hear from Bernat about their new program in our portfolio, which focuses on carbapenem-resistant Enterobacteriaceae and vancomycin-resistant Enterococci. We have CRISPR-phage program that we're also going to hear about today from Xavier. And finally, we have one small molecule program focused on C. difficile in our prevention portfolio. Next slide. Our programs are distributed across the stages of discovery and early development. We end, as I mentioned, at the completion of a first-in-human program. And it's important, of course, to have programs at every stage of development to best ensure success to move to advanced development. Next slide. Finally, I just would like to end by saying a year ago or so, we constructed an internal R&D program or team. And the idea there was to be able not only to support the individual programs, but also to look across them and ask if there are ways that we can help the portfolio and in fact the ecosystem at large. And so we've identified a number of these and are calling them cross-project opportunities. The goal, of course, is to create efficiencies across many of these companies, create a higher level understanding of issues that face these companies, and also to move them forward, even perhaps through a funding call if necessary. I'm not going to go through all of these because I think I'm over my time, but let's say that we have programs that are emphasizing important questions and concerns in all areas of our portfolio. Next slide, please. Finally, for those of you in portfolio, in case you haven't seen your alliance leads, here they are. It's a great team, starting with Richard Alm at the top left in therapeutics portfolio, Ruth Appleby in diagnostics, Ed Berman, largely in prevention, Sue Chang, therapeutics, Nadia Cohen, prevention and therapeutics, Betsy Wonderly Trainer, diagnostics, Maria Urea Nicholson, therapeutics, and all of this is possible because of the hard work of Sarah Walker, our coordinator. Thank you very much.
Hey, thank you, Erin. And for the record, you are not running behind time, so thank you for doing that. Just a reminder to everybody: if you have any questions, please click at the bottom for the Q&A symbol and type them in, and we'll try to get to them at the end of our presentations. Now, it's my pleasure to introduce Xavier Duportet. Xavier is the co-founder and the CEO of Eligo Bioscience in Paris, France. He's joining us today from Paris. CARB-X is supporting the development of their early-stage CRISPR-based bacteriophage that aims to kill these antibiotic-resistant bacteria before an infection has a chance to occur, particularly in the first indication in the gut of an organ transplant patient. He is something of a youthful rock star in entrepreneurial science. The MIT Technology Review named him as a leading French innovator under 35. 2017, Forbes named him as a top young entrepreneur under 30. I think he barely no longer qualifies for an under 30 award. In 2018, he was named Global Young Leader of the Year by the World Economic Forum in Davos, amongst many other honors. He's established a nonprofit, which he calls Hello Tomorrow, which provides support to science industry entrepreneurs in 45 countries around the world. So I'm pleased to present to you, Xavier, and we'll talk about Eligo.
Thanks for the introduction, Kevin. So I'd like to thank you first, you and the whole CARB-X team for making this happen, for supporting Eligo, and for giving us the opportunity to take part in this webinar. Today, I'm going to describe one of the platform technologies that we're developing at Eligo, and that is used for our EB004 program. And as you said, EB004 is Eligo's CARB-X supported program, whose goal is to prevent lethal antibiotic-resistant infections in solid organ transplant patients by delivering a CRISPR-Cas system to targeted bacterial population of the patient's microbiome. Next slide, please. So first, let me start with a quick introduction of Eligo. So I co-founded the company with Professor Timothy Liu from MIT in Boston, but also Professor Luciano Marafini, one of the seminal inventors of CRISPR-Cas engineering at Rockefeller University, and with David Bicard, who is currently our CSO, but also leads the synthetic biology group at Pasteur Institute here in Paris. We are now a wonderful and international team of 38 Eligonautes, all based in Paris, and are supported by leading investors in the microbiome space, including Xavier, a European fund, and Costa Venture in the Valley, but also by public organizations, including CARB-X and the European Commission. Next slide, please. So the human microbiome is a robust and resilient community of over 250 bacterial species engrafted in a variety of ecological niches in our bodies. Altogether, they carry more than 8 million different genes. Host genetics and environmental factors can strongly influence the human microbiome, sometimes transforming its beneficial gene repertoire into a deleterious gene repertoire, which can lead to the emergence or the escalation of disorders and diseases such as infections, but also inflammation and autoimmune disease, among others. And today it's really an exciting time to be working in the microbiome space, as recent advances in metagenomics are now enabling us to understand the causality between the presence of a specific bacterial species or even specific genes within the species and the disorders. At Eligo, we believe that there's a great opportunity for targeted approaches that actually address the precise microbial genetic components of this disease, as opposed to broad spectrum agents like antibiotics, which indiscriminately target a large number of species. Next slide. Eligo is developing modalities that can change the composition of the microbiome through the precise removal of deleterious bacterial genes that do play key roles in the emergence or the progression of disease. To achieve this, Eligo has harnessed the biology of bacteriophage, which are natural predators of bacteria, to create phage-derived particles devoid of any phage genomes, basically some empty shells in which we can package a completely synthetic DNA payload that carries therapeutic genes of interest. These phage capsids can retain their targeting and injection capability that we actually leverage to efficiently and precisely deliver our synthetic therapeutic DNA payload to the bacterial population of choice in a patient's microbiome. This gene therapy of the microbiome enables us to restore a healthy microbiome state without any collateral disruption of the commensal bacterial population. Next slide. A strategy to do this is actually by delivering a Cas nuclease. Back in 2014, when the company was still at the ID stage, we demonstrated for the first time that the delivery of a CRISPR-Cas nuclease system inside bacteria could be used to create a wonderfully efficient and precise antimicrobial. Until then, the ability of CRISPR-Cas to create double-strand breaks in genomic sequences had been used for editing human cell genomes. But we showed that actually using a Cas nuclease to target a chromosomal gene in a bacteria actually led to the rapid death of the bacteria, which is likely because bacteria do not have an efficient mechanism to repair double-strand DNA breaks. We also showed that if the nuclease is expressed, but the target sequence is not present in the bacteria would survive. We had therefore engineered the most specific antimicrobial ever that we named Eligobiotic, that could discriminate the harmful bacteria at the gene level within a complex microbiome, while leaving the rest of the commensal beneficial bacteria completely intact. Next slide. So to put things into context, imagine that you have a mixed population, for instance, of E. coli in a patient's microbiome, with some commensal beneficial bacteria in blue and some that carry a toxin gene or an antibiotic resistant gene, for instance, the red bacteria. The phage capsids carrying the Cas nuclease are Eligobiotic, programmed to target these specific toxin genes, could be given orally, and then would bind and inject its therapeutic payloads in all the E. coli strains, both the beneficials and the pathogenic one. Next slide. The nuclease would then express in all these strains, but only create a double-strand break in the toxin gene, carried specifically by the pathogenic strains, resulting in the rapid killing of only these strains. Next slide. The beneficial commensal bacteria would then be able to take over the metabolic niche occupied previously by both the pathogenic and the commensal strains, and therefore exert a selective pressure on any potential pathogenic survivors, and eventually restoring a healthy microbiome. Next slide. We have demonstrated activity up to 4-5 log killing efficiency and specificity of the approach in a number of different bacterial species and hundreds of different clinical isolates with different gene targets, toxin, antibiotic resistant genes and others, but also different nucleases. Next slide. We have also demonstrated the efficacy of the approach, not only in vitro, but in vivo, by efficiently and specifically decolonize the gut of mice from pathogenic E. coli strains embedded in a complex microbiome and a complex environment, such as the mucus. On the graph on the right, you can see the effect of an oral gavage of our Eligobiotics on the colonization level of a pathogenic strain. The red line is the treatment versus the green line where it's not treated. And the effect being dose dependent over time. And we have now just gathered recently some exciting rabbit data confirming the potential of this approach in larger animals. Next slide. One of the exciting translational projects that we're pursuing with this technology focuses on preventing multi-drug resistant infections in solid organ transplant patients. These infections are actually a leading cause of morbidity and mortality in this patient population. Up to 70% of transplant patients infected with an antibiotic-resistant E. coli or Klebsiella post-surgery die of this infection. It's a very impressive number and sad number. Most of the time, these bacteria are in fact already present in the microbiome of these patients prior to the transplants. But they are kind of commensal organisms or opportunistic pathogens, meaning that they're not harmful for the patient by themselves while they are only colonizing the guts. But it's when they enter the systemic circulation or organs after the surgery that they need to be treated with antibiotics. However, when these bacteria carry an antibiotic-resistant gene, then the treatments likely fail. And so we saw there a real opportunity to try to solve this unmet need with our technology. Next slide. And so the idea behind this EB04 program, which is supported by CARB-X, and is actually to develop an eligobiotic that will be delivered as an oral treatment to specifically remove the ESBL and carbapenem resistant genes from transplant patients prior to surgery. The phage-derived capsid would deliver in this species, E. coli and Klebsiella, a nuclease that is programmed to target all possible variants of these genes, of these resistant genes. So we have demonstrated that actually if an antibiotic-resistant gene is carried on the chromosome by this species, the nuclease-mediated double-strand break would lead to the targeted killing of the bacteria with very high efficiency. What's interesting is that if the resistant gene is actually carried on a plasmid, which is often the case in such strains, then the double-strand break would lead to the curing of the plasmid from the population, which can actually also lead to the death of the bacteria if this plasmid is maintained in the bacteria by the biotoxin and antitoxin system. In any case, the treatment would specifically delete these genes from the microbiome of the patients by killing the bacteria or curing the plasmids carrying these genes, and therefore resensitize the whole population to antibiotics. This strategy will therefore not only not reduce the infections post-transplant, but instead it will reduce the chance of these infections to be caused by antibiotic-resistant bacteria, hopefully significantly increasing the survival rate. Next slide. So beyond this EB004 program, we're using this CRISPR-based killing technology to address unmet medical needs in the anti-infectious space with our lead candidate targeting toxigenic E. coli. This candidate has actually just received orphan drug designation status, is planned to enter the clinic next year, and is backed by the European Commission. In our pipeline, we also have another program focused on selectively removing pro-inflammatory strains from the skin microbiome to treat acne vulgaris. And finally, we have two other not yet disclosed programs in the field of autoimmunity and inflammation. To conclude, we are so thrilled to be working hand in hand with the CARB-X on this exciting program, EB004, and are very much looking forward to bringing to patients an elegant and innovative solution to curb the impact of antibiotic resistance. Thank you.
Thank you, Xavier, for that. And I have to say we're running right on time, so we have no problems in that regard. That was a great project. It's a wonderful example of something that's highly innovative, a new way to address antibiotic-resistant bacteria. Thank you for that. Our next speaker is Jonathan Steckbeck. He's the founder and CEO of Peptilogics Biotech in Pittsburgh, focusing on computational design to discover novel peptide therapeutics. Jonathan has led Peptilogics development into a clinical stage biopharmaceutical company, raising more than $28 million in combined equity and non-dilutive capital to support their pipeline and their discovery platform. In September, Peptilogics received FDA orphan drug designation for its first-in-class broad-spectrum antibiotic peptide to address biofilm infections, which is funded by CARB-X. I give you Jonathan Steckbeck.
Thanks, Kevin. Thanks to everybody at CARB-X for the support for our programs, but also for allowing us to be here today. So next slide, please. So at Peptilogics, we focus our internal development pipeline on anti-infectives. And so like here, everybody here and watching, our team really understands and feels deeply the need for new antibacterials in the face of the resistance pandemic. But we also appreciate that antibacterial development is hard and that the current economics make, successfully developing antibiotics for resistant infections even harder, and really harder than it should be. So that said, for us to be successful as a company, it's been important for us to find a sustainable path toward developing drugs for patients with difficult to treat infections. And so at Peptilogics, we don't develop traditional antibacterials, and our drugs have different activity characteristics than we think of as antibiotics. And so this is important because the activity features of our molecules allow us to take a different strategic approach and focus on important indications that haven't been served or addressed sufficiently with typical antibiotics, which will help differentiate our products upon approval and ultimately support use. And so this strategy of matching technological characteristics to indications where those characteristics confer a specific benefit and let us take a new approach to treatment has allowed us to open up additional opportunities, such as Kevin mentioned, receiving orphan drug designation for our lead CARB-X supported program, which is currently rare in the antibacterial space. Next slide, please. So to give a brief overview of what we do and explain how we arrived at our lead molecule, which was really a design-based approach, and it set the standard for how we approach drug development, which is designing drugs for a specific purpose. So briefly, the class of molecules that we're developing, which are cationic peptides, were discovered 30 years ago, but have been really challenging to develop clinically, largely because of tox problems associated with the molecules. And so our approach to developing this class was to use sequence design to really decouple activity from tox based on an understanding of the molecular SAR. And our goal was to design in the antibacterial activity that we wanted while mitigating what we expected drove the tox by using completely synthetic sequences that had no homology to many of the naturally occurring sequences. And so we started by putting all our early effort into proving that the approach would work and that the molecules are effective and safe. Next slide, please. And so, just to give you an idea of where we started as a company and where our pipeline was when we entered the CARB-X program, and just for some specific clarity here, because it's going to come up later, in addition to the CARB-X support for our 206 program for PJI, we also are running a phase 1 systemic safety trial alongside that to supplement what we're doing with CARB-X. And so, as I mentioned, we're focusing our lead on, we're focused on developing our lead for an indication that really targets an underserved population. And so at the end, I want to contrast this with where we are now based on the support that we've received from CARB-X. Next slide, please. So we're developing 206, as I said, as an orphan designated therapeutic for prosthetic joint infections. And, we're doing this because this is an indication that goes back to our strategy where the current standard of care is, remains aggressive surgery and there's no approved or effective therapeutic. This leads to a really high failure rate, which then leads to a really high, 25% 5-year mortality rate, which incidentally is higher than either breast or prostate cancers. And that leads to a downstream very high cost of failure. And so what we're looking to do is change the treatment paradigm here by providing the first effective therapeutic for patients with PJI. And so this, again, comes back to the strategy that we have as a company where we, are able to strategically create and capture value with our pipeline by focusing on finding these indications, by coupling, the technology characteristics to areas where there's high unmet need and poorly effective standard of care. So we can run trials to demonstrate superiority. In this case with PJI, what amounts to a placebo-controlled trial because the standard of care comparator is saline. And so we looked at PJI as presenting an opportunity for 206 because the existing drugs didn't effectively control the infection. And the challenge was that you needed a drug that could address biofilms, which had proven to be resistant to all approved antibiotics. and that could be delivered and effective during the currently performed surgical procedure to be able to enable those placebo-controlled trials, which required very fast bacterial inactivation. Next slide, please. So to that end, and without going into too much detail, I wanted to give a glimpse of why we and KOLs in the space are so excited about the development of 206 for PJI. So on the left, we can see a rapid, we were able to demonstrate the rapid activity of 206 against in vitro biofilms on implant materials. So we have as a time course of cefazolin, which is a typical standard of care antibiotic for the treatment of PJIs, compared to 206. And what we can see is that 206 rapidly inactivates the biofilm bacteria within 15 minutes of exposure, while cefazolin demonstrates incomplete inactivation with up to 24 hours of exposure. So this is the first time that we were able to demonstrate that 206 has the necessary activity characteristics for success in this space, which was a rapid ability to inactivate biofilms. And then on the right, the question was, would it work in a clinically representative animal model? And so we did these studies with the support of CARB-X. And so here, it's a very clinically representative animal model. We take an exposed pin, sink it into the bone, allow the infection to go for 48 hours, and then compare standard of care, which is surgical debridement with a saline irrigation followed by systemic antibiotics, to surgery with a 206 irrigation followed by systemic antibiotics. It's a very aggressive model. It's 100% fatal, even on standard of care, where, as you can see, all animals receiving standard of care died by day 14, which is typical for the infection. In contrast, 60% of the animals receiving 206 survived to day 28, which was 3 weeks following the cessation of systemic antibiotics. This is the first known cure of animals in this model, and we were able to do it with 206, recreating the human clinical procedure. Next slide, please. And so, as I mentioned earlier, alongside this, to demonstrate the safety of 206, we performed a phase one human study. And so, just showing the idea here, the results, high-level results of our phase 1 IV study, where we've now been able to demonstrate systemic safety and a clean safety profile in humans, along with strong and predictable PK with a 16-hour half-life, which is really rare for unmodified linear peptides. And so what we were able to do, combining this data with the CARB-X data, or the data supported by CARB-X, is really this idea of developing for the PJI application now gives us a really good safety margin. So we're looking at a greater than 500-fold for the treatment in our patient trials, where we will be, as Erin said earlier, treating locally without much systemic exposure. Next slide, please. And so based on our progress and support from CARB-X, I thought it was important to compare where we're at now as a company with where we started the program. We've been able to generate the data necessary that really gives us confidence in our approach and has now allowed us to expand our anti-infective pipeline by leveraging existing data to then expand into other disease indications, all while trying to maintain our strategy of finding areas of high unmet need with poorly effective standard of care, and where our drug, supported by the data, allows us to take a novel approach to providing new therapies to patients. And so going forward, we're going to continue to expand our pipeline with new assets for other underserved indications using our deep learning-based computational platform that Kevin mentioned at the beginning, all while maintaining a strategic focus on identifying indications with really high unmet medical need where we can provide benefit with novel drugs. Thanks, everybody.
Thank you, Jonathan, for that. I really appreciate the presentation. Our next speaker is Dr. Bernat Olle, co-founder and chief executive officer of Vedanta Biosciences here in the Boston area. He's been on the founding teams of several pure tech companies and serves as a member of the board of directors of Vedanta and also Eligo Biosciences. He's won numerous awards, including Innovator of the Year and MIT Technology Review for Spain for innovators under 35. He's originally from the Republic of Catalonia, and he earned his MS and PhD in chemical engineering practice from MIT, where he developed a novel method of large-scale bacterial culture. He also got an MBA from the MIT Sloan School of Management. CARB-X has supported 2 of Vedanta's microbiome projects, one of which has been awarded a $77 million follow-on support from BARDA for completion of the phase 2 clinical development as a preventative therapy for serious recurrent C. diff infection. A second Vedanta project is currently in the CARB-X portfolio, VE707, a microbiome project being evaluated as decolonization approach for CRE and ESBL producers as an infection control strategy for transplant patients and other highly susceptible patient populations. So Bernat, thank you so much for joining us today. We look forward to hearing from you.
Thanks, Kevin. So what we do at Vedanta is we're developing a new type of drugs that are based on defined consortia of bacteria. What we mean by that is that the active ingredients in the drug are live bacteria, multiple different species that need to work together for a common goal. That's why we use the term consortia. And by defined, what we mean is that these products are made starting from pure clonal cell banks, not relying on fecal donations from donors. In the next slide, I have a representation of the key problem that we're trying to address in infectious disease and in infection control. So looking at the left of this slide, a common problem in our healthcare system today is the combination of two factors: high-risk patient and antibiotic pressure on the microbiome of that patient. Patients that are in the ICU have had extended hospital stays, liver disease, kidney disease, bone marrow transplants, the elderly. All of those fall under the high-risk category. And many of these patients, as part of their treatments, the management of their conditions in the hospital setting, have to receive broad-spectrum antibiotics like second and third generation cephalosporins, fluoroquinolones, etc., which exert strong pressure on the microbiome of these patients. Many of them carry C. difficile and other gram-negative bacteria that can bloom after the antibiotic pressure and go on to infect. And that colonization precedes the infection. We think actually this is a bigger problem than the emergence of new resistant strains altogether, which is a very worrisome event, but it's relatively rare. On the other hand, patients that are high risk receiving antibiotics happens every day in every hospital. And once the microbiome resistance against blossoming of these pathogens is lost, it's much more likely that this patient experiences an infection and can transmit this organism in the hospital setting. And so what we're trying to do with V303, with V707, which are two candidates that have been funded by CARB-X that I'll tell you about, is help these high-risk patients reconstitute a normal microbial flora that can fight carriage of these problematic organisms during the susceptibility window that opens right after they get a broad-spectrum antibiotic. Basically, we want to shut down this window of susceptibility as quickly as we can. In the next slide, we have a snapshot of our current pipeline. We have programs in IBD, cancer, allergy, but today I'll only focus on the infectious disease work, which includes our V303 program and C. difficile. That's currently in a phase 2 randomized placebo-controlled study. We received funding from CARB-X that helped advance this program to phase 1. Out of this phase 1, we learned about the safety and PK of the product. We chose a dose that we then moved into a phase 2 study. And recently, we've become a BARDA contractor to continue to advance this product to approval. I believe this is the first time that BARDA has bet on a microbiome life biotherapeutic product. And our ultimate goal is someday for this type of product to be part of the national strategic stockpile, with the use in national security being, or the potential use, that in the context of a pandemic, like the one we're living in now, or a bioterrorism attack with an agent like anthrax, there's an expectation that antibiotic use may need to increase very significantly, which can then in turn lead to a second wave of secondary bacterial infections that follow the antibiotic use. In a way, a second punch. So what we're trying to do is prevent that second punch by coming in after an antibiotic and reconstituting the microbiota. And C. difficile is a perfect example of that. Then separately, we have V707, which is another defined consortia that we're sort of an active student of the CARB-X program for. And there we're advancing this defined consortia to a first clinical study, and we're trying to decolonize CRE and ESBL in high-risk populations. The next slide, there's a high-level representation of how our drugs work. The patient gets capsules that contain a lyophilized powder that has equal amounts of each of the different species of bacteria that make our product. For example, in our CDFSL program, there's 8 different species of bacteria that make up the active ingredient in the capsule. This is ingested orally. The capsule passes through the stomach, reaches the intestine, opens and releases the lyophilized dormant bacteria in the intestine, which then wake up in the anaerobic environment and start colonizing and being metabolically active. We've shown in humans, as I'll show you in a following slide, how these organisms can colonize very abundantly and durably in the intestine, and that's a critical part of their mechanism of action. If we give them dead, they don't work. They have to be alive. And also in a different time frame, they can stimulate a range of immune responses, which we explored in other indications like IBD and cancer. The next slide. This is a brief explanation of how what we're doing is different from what has already been done in the microbiome field. Fecal microbiota transplants have already been demonstrated useful in the field. Spore fractions of fecal microbiota transplants have also shown utility in phase 3. And here I use the analogy of what happened in the blood products field to explain where I think the microbiome field is going to go. So in the whole plasma field, we started with plasma donations earlier in the century. Those had some safety issues, as fecal transplants do as well, with the potential risk of transferring pathogens like bacteria and viruses. Some of those issues were addressed by treating plasma with ethanol, which kills many of the viruses. This is also being done in the microbiome field via treating fecal transplants with ethanol to get the spore fractions. And that increases the safety, but it doesn't eliminate all the potential pathogens. But ultimately, what characterizes all these donor-derived processes, whether you're donating plasma or feces, is that it is a process. It is not a product of standardized composition. And so what we're trying to do in the microbiome field is the equivalent of what the biotech industry did with blood products. We reached a point where the industry figured out how to create clonal cell banks, which obviated from there on the need to rely on donors of unknown composition, and therefore gave the ability to control the supply and the safety of those drugs. And since then, we've had more than 100 monoclonal antibodies approved, and the industry never looked back. In the microbiome field, we're trying to do the same. We're starting from products made from pure clonal cell banks, which means we know exactly what we give to the patients, and we know what we don't give them. And we can also do that via a process that is scalable. In the next slide, we have a little bit of data from our CDFSL program, including the data we've generated with CARB-X, showing on the upper left in animal models that we could find a defined consortium that was equally active to a human FMT. On the lower left, going into human data, that these organisms can colonize very abundantly and durably in patients, the order of 10 to 100-fold higher than probiotics, and with durations that can go out to a year. We've also seen upper right that the multiple doses do better than single doses, and also lower right that there's a pharmacodynamic effect on the resident community of bacteria in the host, where we help recover beneficial bacteria in the host and eliminate problematic bacteria. And then my final slide, I want to put a word on the V707 program, which is currently an active program with CARB-X. And here what we're trying to do instead is to colonize CRE and ESBL. A number of populations that are high risk, like recurrent UTIs, ICU populations, stem cell transplants, get colonized and infected with those organisms. Antibiotics have been used to show proof of concept, but of course the issue is emergence of antibiotic resistant strains. And FMTs have also shown proof of concept, but with great variability from donor to donor. So our effort here is to create a standardized, defined product that has reproducible potency across the patient populations of interest. That's all. Thanks for the time.
Thank you. All three of you did very well in your time. We're ahead of schedule, which is excellent. I have to say that your 303 project is the second CARB-X funded project to receive follow-on funding from BARDA. The first was a diagnostic from T2. We're grateful for that. We want to see a lot more of those, and looking forward to the Action Fund standing up and taking some graduates as well. We have several minutes for questions. We've received, I think, 13 questions at this point. You can add additional questions if you want down at the bottom of the panel. But I'm going to start with Xavier. There's 3 questions I'm going to combine. One is about the 98% of the target population. You know, do you need a combination of phages to get there? The second question was, what sort of resistance are you seeing to the phage? Even with the Cas nuclease. And then finally, how does the phages, when it's coming in an oral sort of treatment, how does it survive the harsh GI tract? If you could address those, then I'll bundle some questions for Jonathan next, and then.
Sure, happy to. So for the 98% targeting of the gene variants, the beauty with this with the CRISPR-Cas approach is that actually in the therapeutic payload you can add different CRISPR guide RNAs that you can multiplex, and so combine different targets within the same payload to make sure that actually if you have variants, each design target will actually cover one specific region that is homologous in that is common in maybe 50% of the variants. The other guide that will target other sequence that are common in the rest of the 25%, and you go further. And so we've demonstrated that we can add up to now we can combine 6 different guide RNAs on the same payload, and this is enough basically to cover about 98% of the different variants. Then in terms of so how to get the how do phages so phage capsids survive the harsh gastric passage. So I think we can phage can be lyophilized, phage capsid can be lyophilized and then put into a lot of, you have different type of coatings and strategies to actually make sure that what's inside these the pills bypass the stomach and can be delivered at different places in the gut. And so right now we've actually also demonstrated that this works with hydrogel. Our lead candidate is using a hydrogel, and we have demonstrated that both in animals and in simulated guts that we could have delivery, safe passage of our capsid and delivery at the right place in the whether it's in the small intestine or the colon. And then on the question for resistance, so actually, so there are different type of resistance that our oligobiotics encounter. I think as opposed to lytic phages, where resistance are first on the receptor. Bacteria can mute the receptors, and therefore the phage cannot bind anymore. So we face this one too, and for this we can actually engineer, the tail fibers, what binds to the bacteria, to actually have multiple binding capabilities. And we can also, we are also exploring doing a cocktail of different capsids to make sure that you can target different receptors to bypass this restriction, these mutations and resistance. But then you have much less resistance in terms of internal mechanism of the bacteria as opposed to lytic phages, because for lytic phages to kill the bacteria, you need not only to inject the genome of the phage, but you need then you need to mount an infection, a lytic cycle within the bacteria to replicate the phage, and you have a lot of way for the bacteria to kill the phage there to prevent this from happening. On our side, we just need to inject the DNA, express the nuclease, and then it kills really fast and very efficiently. One of the resistance that can happen actually is that it's not really a resistance, but you can have a mutation in a target sequence, and then the guide RNA cannot really find the right sequence and cleave it. But again, here, this is why we're actually using multiplexing, where we add different redundant guides that can target different sequence within the gene to make sure that actually, if one of the target sequence has a mutation, there will be another sequence targeted to still kill the bacteria.
All right, thank you for that. There's a lot of questions for Jonathan, and just to highlight a couple of them. What about recurrence? I guess if all the rabbits are dying, then it's hard to assess recurrence at this point. Have you assessed the frequency of in vitro resistance? What is the microbiological spectrum of the activity of your modified peptide? And do you know if people are raising an immune response? There were more, but we'll stick to that for the next 5 minutes.
Sure. So the microbiological spectrum, that's a relatively easy one. So the peptides are broad spectrum agents. So they're effective against both gram-positive and gram-negatives, which, in the context of PGI is important because, it's really an empirically treated infection. So the surgeon doesn't know ahead of time going in, what the causative agent is. Now it's often staph, but particularly in recurrent infections, it can be enteric gram-negatives as well. So, one of the benefits of 206 is that it's a broad spectrum agent able to handle both, all the gram-positives or gram-negatives that are recurring in the infection. And then the other question was around frequency of resistance. Yes, we have tested frequency of resistance. And so we see a very low frequency of resistance on the order of 10 to the minus 9th, 10 to the minus 9th for 206. And then, sorry, Kevin, what was the other question?
About the rabbit model?
Yeah, the rabbit model. Yes. Yeah, so I think, as Kevin mentioned, it's a pretty aggressive model where all of the rabbits treated with standard of care die. Now on the surviving animals, so the animals treated with 206, they were off of antibiotic treatment. So this, so the treatment is a single treatment with 206 at the time of surgery. And then in this particular instance, they received 5 days of systemic cefazolin, which was then stopped. And so then the animals were carried for another 21 days. So the expectation is, if you kind of go back and look at the curves with standard of care, as soon as the antibiotics were stopped in animals that had a saline irrigation with cefazolin, by five to 7 days later, they were all dead. Whereas the animals treated with 206, there were 60% of them carried through for 3 weeks off of antibiotics. So the expectation is that if there was a recurring infection, that it would have essentially killed the animals during that intervening time. At the end of the study, we did do, upon necropsy, we looked for live bacteria or evidence of bacterial infection in the joints. The tissues all looked clean. The cultures came back negative, so it appeared to be sterilizing, or as close as we can tell, a sterilizing type treatment.
All right, we have a number of questions that are directed towards CARB-X, but I'm going to pause on those. We can always put those together and put out a Q&A document, an FAQ document. So for Bernat, tell me that there's two things I'd like to direct your way. One is, what is the endpoint for a trial on CRE eradication? How are you going to define that? What sort of, you're going to look for a microbiological endpoint, a reduction in infection, reduction in harm from infection, what's your goal? And a broader question from that is that, a lot of these, really for all three projects, the clinical trials will require a clever design, and careful consideration. Maybe you can shed some light on what you're thinking in that regard.
Yeah, this is a very important, perhaps the most important clinical development question, because how feasible it is to conduct a trial is quite different if the endpoint is decolonization, which would be a proof of mechanism, versus if the endpoint is infection or mortality, which is the more clinically relevant endpoint, but also makes the recruitment and the execution of the study much more difficult, unless, of course, you can identify a patient population where you have a very high rate of colonization, then resulting in infection, in which case the numbers of patients that you need in the treatment versus control arms may be manageable. So I think it's really, first of all, establishing proof of concept by showing decolonization, I think is a worthy outcome, because it will help us understand how the drug works. And my personal view is that in some of these conditions, decolonization is absolutely strongly correlated with better outcomes of infection. And this is something that is strongly believed by some of the academic leaders in the microbiome field. Whether the FDA will agree with that or not will take more discussions. I think ultimately a product that is approved and broadly commercialized will have to show an impact on infectious outcomes and mortality. I think that the question we'll need to get better clarity on is how much use and mileage can you get of a surrogate marker endpoint of decolonization.
Great, thank you. There's a number of questions that are directed towards CARB-X, and Erin, I'm going to put one to Erin at the top there. Nia Prasad asks, antibiotics don't normally get orphan drug designation. In the cross-project analysis, a strategy has been identified, for all these regulatory pathways. Maybe I can invite you to comment on anything that's been spoken of before, but also to talk about what you hope to achieve with cross-project opportunities.
Sure, Kevin. So again, a number of the cross-project initiatives are focused on technological gaps or challenges that companies face. So in that list that I showed, things that are underway, we have a program that we're actually conducting with one of our product developers that's focused on nephrotoxicity, which is, of course, something that has plagued a lot of our programs, in particular, or programs in the general antibacterial space, including peptide programs, but other classes as well. No one really knows what the right in vitro or in vivo correlates in the clinic are. And so we've undertaken a unit of work, not only to define that, but also to do some back translation of molecules that have been in the clinic or used on the market. There are other programs in the therapeutic space focused on susceptibility and pre-existing pools of resistance, in the prevention space focused on antigenic conservation. And so these are just getting underway as well. The question about regulatory path and clinical trial design is something that we think about a lot, particularly with programs like you've heard from today, all of which are really trailblazing in a way, both scientifically and regulatorily, if that's a word. And one of the things we want to do is to engage regulators both in the US and abroad, both in the drug and the biologics arenas, in clever ways, maybe through public calls, maybe through small group settings, to really have conversations about clinical trial design and paths. We also would like to engage with clinical trial groups like the ARLG here in the US, who may have interest in helping us to move some of these programs forward.
All right, we have 2 minutes left. There's a lot of questions that are not yet answered. But one near the bottom is from Ariel Blocker asking, did I understand correctly, after you announced project number 75, CARB-X will take a funding break until we apply and obtain new funding from the US? The number 75 isn't exactly correct. We have a number of programs that are under negotiation. So the final number of programs may be, in the mid 80s. It'll depend on what comes across the line at the end of the day. But essentially, yes, until we're able to secure additional funding from the US government, we won't have entirely new rounds, because we've reserved the funding that we have to make sure that all the projects that we currently support, we can support adequately all the way through graduation from CARB-X after their phase 1 work is done. Erin, there was another question on here for you. I don't know if you want to pick that up or.
Is this the question about the program that targets AGRA?
Yeah.
So the program that targets AGRA is the BV200 program of Bioversus.
All right, so we're sorry we couldn't get to all the questions. There's still 7 or 8 of them left. Some of them are quite specific to specific companies, and I'm sure you know how to follow through with these folks. Thank you so much for your time.